Dr. Ansuman Satpathy comes to us from Stanford. He got his MD and PhD degrees at Washington University, then joined Stanford, did his clinical residency there, did a postdoc in epigenomics in the lab of Dr. Howard Chang, and currently works on both single-cell and three-dimensional genome sequencing with respect to understanding the epigenetic regulation of tumor-specific T cells. So thank you very much, Dr. Satpathy, for joining us.
So thanks really very much for the invitation to come here. To the organizers, really a pleasure to be here. I've been using BD products for the last 15 years, so it's really nice to visit and see the place. So I think what I'm going to talk about today is slightly different than what the previous speakers have talked about, but hopefully by the end of the talk you'll see the relevance, and we'll bring it from sort of basic genome technologies back to clinical applications. So what I'll tell you about today is really focused on developing genome technologies, in particular technologies that measure epigenomics, and I'll tell you what I mean by that. So what our group is generally interested in understanding is how regions of non-protein coding DNA in the genome impact the expression of disease-associated genes. And when we talk about the non-coding genome, which makes up about 98% of your whole genome, we're talking about primarily two things. We're talking about cis inputs, typically thought of as enhancer regions in the genome that control gene expression, or trans inputs, which are typically thought of as transcription factors that bind to these elements and bring them close to the genes that they regulate. Okay, and why do we care about this? The reason that we care about this is that if you look at, over the last 10 or 15 years, a number of genome-wide association studies in a wide array of diseases, about 95% of the disease risk actually falls in this non-coding space. Okay, only 5% in actually the protein coding genes. So clearly there's some interesting information here. There's some molecular regulators, some molecular switches that get changed in the setting of disease and cause a disease phenotype. Okay. So how do we understand this, and how do we actually measure this in primary cells? And the challenge is essentially twofold. Okay? The first challenge is to identify where in the linear genome do these regulatory sites fall. Where are the enhancers? Where are the promoters that control gene expression? And let's say we can even meet that challenge of finding where they are and doing that in primary cells and doing that in patients. These sites can actually be hundreds of kilobases to even megabases away from the genes that they regulate. Okay? So how do we now map these enhancers, which are megabases away in the genome, to the genes that they actually regulate? Okay? So it's more of a three-dimensional challenge. And the problem has been that prior gold standard technologies in the field really required a ton of cells to do this. So hundreds of millions of cells or billions of cells. And so you couldn't do this in primary immune cells, and certainly not in any serial fashion from patients. Okay? And so largely people have done these types of studies for the last 10 years in cell lines or ex vivo expanded cells. Okay? So how do we actually get this to where we can apply it to patients? So what I'll tell you about today is several technologies that we've developed over the last 2 or 3 years. And I'll start with basically, really basic descriptions of the technologies and sort of give you a sense of what they can do. And then at the end of the talk, I'll show you how we're applying them to immuno-oncology and other disease settings. So our group, over the last few years, has developed 2 primary technologies that allow us to now map the epigenomes in primary cells. The first technology is called ATAC-seq, or Assay for Transposase-Accessible Chromatin with Sequencing. This is a technology that was developed by a former graduate student, Jason Boyne-Rostro, in the group, who figured out that if you take this naturally occurring transposase enzyme, so it's a bacterial transposable element enzyme, whose function it is to basically cut and paste DNA across bacteria. Okay? But now we can engineer this protein to have Illumina sequencing adapters, and then just dump that complex enzyme onto either cells or nuclei. And then what will happen is that enzyme will then integrate into open areas of chromatin. Okay? So these open areas of chromatin are essentially enhancers and promoters. And then you can PCR the library, these fragments that come from this transposition, and then sequence to identify really with base pair resolution where are the enhancers and promoters genome-wide. Okay? So I'll come back to ATAC-seq in the second part of the talk, but I think this has really been a revolution for immunologists because you can now take this type of measurement in primary cells. Now you can see what's happening in the other 98% of the genome. So you can do this in 50,000 cells in the sort of the first iteration. I'll show you later we've now taken it to the ultimate resolution of single cell detection. The problem with ATAC-seq is that you can find these elements, but most of them are actually in sort of non-coding space, right, as you would expect. They're not on genes. So how do you then map them to the genes that they regulate? So in the first part, I'll tell you about a new technology that we've developed called HiChip, or H3K27 acetyl HiChip, which allows you to identify three-dimensional functional enhancer promoter contacts genome-wide in primary immune cells. So I'll just briefly take you through the technology. We can talk more in the break or afterwards about the details. Essentially, this is a in situ Hi-C-based approach. So what we do is we take the chromatin in situ in a cell, cross-link it, and then physically ligate pieces of DNA that are interacting together in the nucleus together. Okay? So they could be far apart in the linear genome or close together, but if they're interacting, we link them together. And then we pull down the regions that are associated with K27 acetyl and then sequence these hybrid fragments as a library. So each end of a fragment came from two parts of the genome. We map that computationally to then infer interactions in the genome. So let me show you some primary data. I think it'll become a little more clear. So this is the first HiChip experiment we did. This is in K562 acute myeloid leukemia cells. And we've done this, of course, the data is genome-wide, but we're going to just focus on one gene first, okay, just to show you the proof of principle. So this is the MYC gene. Here's the promoter in blue. And what we're doing is walking down the chromosome and asking where do we see signal with HiChip. So where do we see 3D contacts that are also enriched for active enhancers? And you can see we see 5 peaks, right? So 3 that are relatively close by, about 100 or 200 kilobases away. 2 which are actually quite far away, 2 megabases away from the gene, and actually skip over intervening genes to interact with the MYC promoter. So how do we know this is real? How do we know these are functional enhancers and not just some type of noise in the data? And so what we did in this case and in several others is to compare the signal that we get from HiChip to high throughput functional CRISPR screens. Okay, so these are tiled screens. And so in this case, we compared it to a screen done by Eric Lander and Jesse Engritz at the Broad Institute. And what Jesse and Eric did is they tiled 100,000 CRISPR guides along this exact same locus and asked which of those CRISPR guides impacts MYC expression. So you can see they found 5 guides, so 5 regions that act as enhancers for MYC, and those perfectly overlap with the 5 that we get. So we can reproduce this 100,000 CRISPR guide experiment using a single assay. I'm showing you one locus here, but of course the data, you get the same data for every gene genome-wide in a single assay. Okay, so the point of these first few slides is just to give you a sense of what we can measure and how useful is it, and how diverse is it across immune cell types. Okay, so I'm going to try and convince you that we should actually be measuring this space, and then show you how we've been able to do that in patients. So the first question we had is, do we actually need to do this sort of complicated 3D enhancer mapping in primary cells? Can we just do it in one cell type and infer that it's the same everywhere? Okay, so do we actually need to do this in patient samples? So here I'm showing you the same MYC locus, right? And the same sort of enhancer analysis using HiChip, but now we're looking at a B cell line or a T cell line. So these are relatively similar cells. I mean, I know for immunologists they're very different, but they're both lymphocytes, right? So you might expect that they're, I'm also an immunologist, but just work with me. So you might expect that they might have similar landscapes, maybe some slight variations, but largely similar for controlling MYC. But what you actually see is dramatically different enhancer landscapes, right? Even down to the direction in the genome in which the enhancers go. So T cells colored in purple here, the enhancers go off to the right of the gene, and B cell enhancers go off to the left, right? And we can then validate these enhancers using our own CRISPR-I experiments and show that knocking down the appropriate enhancer in the appropriate cell type leads to loss of MYC expression and not the inappropriate enhancers. Okay, so can we now actually take this to patient samples? So this was the original goal of the method. And so what I'm showing you here is essentially raw HiChip data from 3 starting cell pools, 25 million cells, 1 million cells, or 50,000 cells. This is still done in cell lines. And the plot here is a 2D interaction genome map. So this large diagonal here is each piece of DNA interacting with itself. And anything off the diagonal in a focal spot is 2 pieces of DNA that are distal or far away from each other in the linear genome acting together in the 3-dimensional genome. Right? So the challenge was to take this type of analysis from about 50 million cells to about 50,000 cells, right? So improve the signal to noise by about 3 orders of magnitude. And so you can see we get nice signal to noise in 25 million cells and can maintain that down in 50,000 cells, and we can go as low as about 5 or 10,000 cells. Okay, so having seen that, we now move to primary immune cells. We wanted to study T cells for sort of the variability in phenotypes from relatively similar cells and also for their relevance to immuno-oncology. So what we did first is to isolate various CD4 T cell flavors from the peripheral blood of healthy patients. So these are the, sort of the first attempts to make these sort of genome maps in primary cells, primary immune cells. And so we isolated from a single blood draw, about 3 mils, either naive T cells, T helper 17 cells, or T regulatory cells, and did HiChIP. And you can see that we get these really beautiful genome conformation maps. So what you're looking at as you go from left to right is essentially zooming in on a piece of the chromosome. So you can see sort of higher order chromatin structures on the left going down to individual enhancer promoter connections. Okay, so does the data that we get actually tell us something that's unexpected? Do we get sort of complex 3D interactions that we couldn't have predicted just from sort of proximity to a gene or one-dimensional data? And the answer, of course, is that we get a lot of complex interactions. About 80% of the interactions that we see are some type of complex, unpredictable behavior. So these are things like enhancer promoter skipping, which are essentially distal enhancers skipping over intervening genes to interact with another gene. Enhancer clicks, which are several enhancers working together to regulate a gene. Promoter-promoter interactions, which are promoters serving as enhancers for another gene, or the sort of dynamic enhancer usage that I just showed you in the MYC locus. And importantly, I don't have to tell this audience, but it's really important to do this in primary cells that you care about, right? So interactions that we see, for example, in T regulatory cells, of course you can't see in cell lines using prior gold standard techniques or even in total T cells. You really need to do it in the patient or the cell type that you care about. Okay, so just to end this sort of technology piece, I want to return to the question that I asked in the beginning, which is that can we now use these 3D maps to nominate potential target genes for intergenic SNPs associated with disease, in particular autoimmune disease? So about 95% of the SNPs associated with most autoimmune diseases fall in this non-coding region. Can we then map using HiChIP those SNPs to target genes? Okay, that's the concept. So we'll focus on individual examples first, and then we'll sort of zoom out to global analysis. So what I'm doing here is now plotting HiChIP signal, okay, the same types of plots that you just saw, but now we're centered on a SNP associated with type 1 diabetes or rheumatoid arthritis. Okay, this is a really highly correlated SNP. It's been reproduced in several studies. So actually, this SNP is closest to the gene SMIM20. Okay, and I don't know about you guys, but for me, that didn't really ring any bells. Okay, it doesn't really have a known immune function. But what you can actually see is that, using the HiChIP data, is that you don't really get much interaction between this SNP and SMIM20. But instead, you get really nice interactions with RBPJ, which is a notch regulator, and STIM2, which is a T cell activation gene. Okay, so this makes much more sense with the biology of the disease. Similarly, for a SNP associated with multiple sclerosis, it's closest to this gene RMI2, but actually makes very nice interactions with SOX1, a regulator of cytokine signaling, and CLEC16A. Okay? So we can now zoom out and do this for about 21 autoimmune diseases where we have nice GWAS data and expand our list of nominated gene targets by about 4-fold. And importantly, only about half of the nearest gene to those SNPs actually showed any appreciable signal. We can even go a step further and look in patient samples using HiChIP and ask what actually happens to that loop in a patient who has that SNP. Is the loop strengthened or broken, right? And you can see both behaviors in patient cells. So in some cases, the SNP leads to enhanced interaction of that loop, of that enhancer with the promoter, and in half the cases, it leads to breaking of that loop. So as we move forward now into thinking about how can we actually use these enhancers as sort of molecular switches to control behavior or study the pathways of disease, we're taking sort of both approaches to both weakening the loop and strengthening the loop using CRISPR-I or CRISPR-A targeting. So here what I'm showing you is the same two loci that I just showed you associated with autoimmune disease, but now what we're doing is we're targeting a CRISPR interference domain, so the transcriptional repression domain to that SNP instead of just analyzing it. And so what you can see is that by targeting that SNP with a CRISPR repression domain, we're able to dial down the expression of the gene that we predict by high CHIP. So conversely, we can target those SNPs with a CRISPR activation domain and show that we can dial up the gene expression both by protein and by RNA. Okay, so what I wanted to sort of convince you about in this first part is that epigenomics might have sort of a useful angle of measuring in immune cells, right? And that it varies widely between immune cell types and potentially between patients. We've developed a new technique that allows you to now measure this in primary cells. We've done it in T cells, but of course it should be applicable to any cell type that you care about. So I know that probably, I'm at BD, right? So everybody is thinking here, 10,000 cells, that's great, but we really want single cells, right? Immunologists want to study things at single cell level, and that makes sense particularly for T cells, right? Because every T cell has a different behavior driven by its receptor. So can we now use these sort of epigenomic information and integrate it with other things that we've known for a long time about those cell types? So things like cell surface markers, antigen receptors, particularly for T and B cells, even sort of perturbation states, and sort of integrate all of that together to get a really sort of full profile, molecular profile of that cell, right? And so we think about this in the same way as we might think about Google Maps, right? That the reason Google Maps is useful is that you're integrating many different layers of information, right? So you know where the people are, where the streets are, where the coffee shops are, where the restaurants are, and then you can use that information to really navigate your way to where you want to be. So in the same way, integrating epigenomic information might give you a deeper understanding of the cell function, cell state, and how that might last over time, right? So instead of just a snapshot, epigenetic marks are inherited. So why do we think that epigenomics might give us another angle? You could just study RNA or protein. What do we think that we add here? And so here I'll show you a very simple example. And so this was done by Howard's group, Ryan Corses and Jason Buenrostro, who did a TAC-seq analysis sort of through the continuum of hematopoietic development, okay, starting at hematopoietic stem cells, going down through progenitors, and then end-stage cells. And what they found in many, many cases is this type of behavior, where the gene expression, so as measured by RNA levels, is relatively similar, right? So within 1 or 2-fold different between cell types, and even by protein, it's less dynamic than that. So the protein, if you look at intracellular staining, is relatively the same. But what you can see in the enhancer landscape is that you get dramatically different enhancer usage, right? So here's the promoter here, this big peak here. So every cell type has that promoter. But each cell type has its own set of unique enhancers that really vary broadly and widely across this window of the genome. Okay? So the way that, and if you do this, if you use sort of RNA-seq or protein profiling versus epigenomic information, you can get sort of better clustering using the epigenomics, right? So this is clustering of all the immune cell types using RNA-seq. And so you can relatively well separate out cell populations, right? You get some mix-ups and some T-cell subsets. But you get a cluster purity about 77%. With distal enhancer, clustering the cells only by distal enhancer usage, you get much better separation. So the way that we're thinking about this is that epigenomic analysis might give us really an insight into the underpinnings of how a cell gets there, right? So what's the regulation of the RNA and protein expression, and how might that be inherited over time, right? So it gives you really, so everybody knows this Waddington landscape picture where cells are sort of going down cell fates, and we view this as, you can measure where the cell is in this trajectory using maybe protein staining or RNA staining. What many people haven't seen is the next picture in that book, which is essentially the underside of this landscape, right? So these are what's actually holding the landscape in place, what's making the cells differentiate into different ways. So we view the epigenomics as being sort of the underpinnings of what we can read out by RNA and protein sequence. Okay, so can we actually do this? Can we now take epigenomics to a single cell scale and then integrate it with other types of information? So I'll show you 2 examples. One is probably the one that you all are most interested in, which is integrating epigenomic information with some information about the antigen specificity of that T cell. Okay, and so TCR alpha and beta sequence from that cell. So we call this method transcript indexed RNA in a TAC-seq or T-ATAC-seq. And essentially you can do this either using a microfluidic device or single cell sorting, and I'll show you examples of both. but the process is the same. So you capture a cell and then take it through sequential steps that give you ATAC-seq information, and then you move all those chamber contents to a separate chamber where you do TCR sequencing, both alpha and beta paired sequencing. So then at the end of the day, you have two different libraries from the same single cell, an epigenomic library and an antigen specificity library. You amplify both of those either together or individually, sequence, and then computationally integrate. Okay? And so of course I don't need to tell this audience the reason to do TCR sequencing is that you can get a lineage trace of the cell, so you can follow the cell over time in response, for example, in a tumor. And then you can pair that with the epigenomic information, which might identify regulators of that cell state. Okay, so what does the data look like? So this is a genome track where every peak represents an open chromatin site. So it's either an enhancer or a promoter. Okay? The green line is the prior gold standard method, DNase hypersensitivity sequencing, done in 10 million cells. Okay? The red line is ATAC-seq done in 50,000 cells. So you see we get relatively the same profile. And the bottom blue line is aggregated data from 200 single cells, okay, done using this t-ATAC-seq method. So now you can see that we get the same information from about 200 cells as you could previously get from 10 million cells. Of course, now the benefit is that you can see the single cell resolution, right? So you can see now, so in this heat map, every row is a different cell and every column is an enhancer. And so now you can see enhancers that are constant across T cells or variable in this population. Right? And then of course for every single cell we also have TCR alpha and beta sequence. Okay, so now we've done this in primary cells and we're going to focus again on the same cell types that I showed you high chip data for. So these are CD4 T cells sorted from the peripheral blood as using sort of canonical markers for each population. So naive T cells, memory T cells, and Th17 T cells. And now we've taken them through this t-ATAC-seq protocol. And so this t-SNE reduced dimensionality plot is showing you basically the overall epigenomic signature of each of these single cells. They line up mostly as you might expect. So most of the naive cells are together as sorted by protein staining. Most of the memory cells are together and T helper 17s together. You do see a small population, about 5% of the naive cells that actually fall in this memory gate. And we've done some work and others have done work of validating that population. So now, what can we actually see from each single cell? So let's zoom into this one cell here. It's sorted as a naive cell, but clusters as a memory or T helper 17 cell. And now let's look at about 10 to 20,000 individual sequence fragments from that single cell. Okay, so now we can find where are the enhancers and promoters that are making that cell go into this memory cell state. And then we can even go a step further. We can zoom in to each individual peak and look for transcription factor footprints within that peak. So you can imagine this as if you're spray painting a wall and you put your hand in front of the spray paint. When you're done, there's an imprint of your hand, right? So in the same way, if a transcription factor is sitting on top of the DNA and you're transposing that DNA, when you're done, there'll be a little imprint of where that transcription factor was sitting. And so in that way, you can infer what's the transcription factor with some confidence, not 100% confidence. What was that transcription factor that's sitting in that peak? And then you can do that over and over and over for every peak, genome-wide, and get really an idea of what's the overall activity of each transcription factor in that cell. Okay, and so this particular cell has a lot of AP1 motifs, which are associated with TCR signaling and T memory formation. So the nice thing is that you can do this for about 1,000 or 2,000 transcription factors that we have nice footprinting data for. Okay, so now these plots are the same t-SNE plot, but colored by the activity of each transcription factor. So I'll just show you one example which might make sense, which is the motif for ROR gamma T, which is really highly active genome-wide in Th17 cells, but not so much in naive and memory cells. Okay, so that's how we look at the epigenomic data. How can we now integrate this with TCR to do something useful in patients? Okay, and I'll show you two very small examples of that, or short examples of that. The first is for looking at epigenomic signatures of T-cell lymphoma, and the second is looking at the T-cell response to skin cancer in the setting of checkpoint blockade. So we'll focus first on T-cell lymphoma because I think it's the simplest to really understand and walk through. And so the idea is that this might be a good setting to actually do epigenomic analysis, because this is one of the few places where there's actually an epigenome inhibitor approved by the FDA for treatment. Okay? So you can treat these types of tumors. Second-line treatment is HDAC inhibitors. Okay, so can we now use our epigenomic profiling to predict who might respond to these therapies or follow the response or segregate them by by outcomes. So this is a patient that came into Stanford with stage 4 T-cell lymphoma or Sézary syndrome. We couldn't determine the TCR in the molecular lab in pathology, but it had a super high CD4 T-cell count in the blood. So we took this patient's peripheral blood through T-attack, and this heat map in the middle is showing you essentially the universe of all TCR rearrangements. And so you can see that this patient has a really huge hotspot in this particular rearrangement. So about 73% of their T cells have a single TCR. So that's their putative T cell cancer clone. The most you would expect in a normal patient is 1% or even less. And so now the interesting thing that we can do is we can go into that clone and look at the epigenomic signature compared to all of the other non-clonal T cells. Okay? And then do the same thing that I just described to you, look at transcription factor activity or cis elements. And you can see the signature of these T-cell lymphoma cells having high AP1, high GATA1, and GATA3, which sort of makes sense with prior literature for this tumor type. The interesting thing was that this patient did get HDAC inhibitors and didn't respond at all. So we were thinking, what if we can expand this? Can we use this information to predict that therapy response? So we've done this now in about 100 patients that have come to Stanford. And we can find essentially 3 overall epigenomic signatures of CTCL cells. So again, all of these tumors are phenotyped using flow profiling and other things, and largely have the same profile. So you couldn't really determine anything from that. And so indeed, the patients who have, so there are about 20 or 30 patients who had the same signature as this first patient that we saw, and all of those patients did not respond to HDAC inhibition, right? So this suggests that you could use epigenomic signatures to predict perhaps therapy response. The second place that we've done this is now in the setting of immuno-oncology. So looking at T-cell responses to checkpoint inhibitors, okay? And so there's some data around using epigenomics to study T-cell phenotypes. So 3 papers that came out last year from John Wary, Andrea Schiedinger, and Nick Haining that showed that T-exhausted cells, right? So these are cells that see the antigen in a tumor or chronic viral infection and then basically become dysfunctional because they've been overworked. They actually occupy a distinct epigenomic profile than memory cells or effector cells. Okay, so something on the order of 10,000 differential cis regulatory sites. Okay, and what John's lab showed is that this epigenetic state was actually critical for maintaining that T cell in this exhausted state. Okay, so after PD-1 inhibition, the exhausted cells would not get reactivated, and that was because they were sort of locked molecularly in this exhausted dysfunctional state. Okay, so all of that work was done in mice, and so we were thought, with the new sort of resolution that we can achieve with these technologies, can we actually start to do this in patients? So this is a patient who came in with metastatic basal cell carcinoma. So this is a very common cancer, the most common cancer in humans and skin. And actually 95% of these cancers you can just cut out, right? So they're actually high UV mutation burden cancers, similar to melanoma. You can cut most of them out. For the 5% or less that become metastatic, there's not much you can do. There's some biologic treatments, but not much else. Only about 30% of those patients will respond. So now studies have started to do checkpoint inhibition, and about 50% or a little more of metastatic BCC patients will respond extraordinarily well to PD-1 monotherapy. So what we did is look at this patient who came in and actually got PD-1 therapy and was doing well for about 6 months before he relapsed. Okay? And so we now profiled the tumor cells using T-ataxy. And so this plot here is a UMAP, so it's a reduced dimensionality plot of all of the TILs that we got from that patient's tumor. So it's actually about 12,000 T cells. Okay? And so you can see a population of Tregs, some CD4 T cells of various flavors, activated T cells, and then you can see this small cluster of actually exhausted CD8 T cells. Okay? Here. And so now if we can pair this epigenetic information with the TCR profile and highlight where the expanded clones are within that population of T cell. So now in this plot below, the colored dots are an individual clone. Okay, so here's a clone in memory T cells, a memory T cell clone, and then here's an exhausted T cell clone. So a number of studies have come out recently saying that neoantigen-specific cells have the expression of certain markers like CD39. All of these cells have those markers. One thing you can notice is that clones tend to behave similarly, right, in the epigenomic space. So they will largely cluster together, and here are two examples, but this is true across hundreds and hundreds of clones. Okay, so now can we actually try and understand what's locking these cells in this exhausted state? So now we can zoom in to these clonal exhausted cells, and here I'm just showing you the comparison of this exhausted clone against this memory clone, and look at both cis-regulatory elements and transcription factors. And indeed, you can find the opening of some enhancers in exhausted cells that are extraordinarily specific to the exhausted cells and not to memory or activated cells. Okay, so this is the CTLA-4 gene, which you all are familiar with. CTLA-4 actually is induced in T-cell activation, other states, right? So the expression of CTLA-4 in exhausted cells is not specific, right? But what is specific is the opening of these very specific enhancers that are about 100 kilobases and 20 kilobases away from that gene. So somehow the exhausted cells are using a different molecular wiring to control the same genes, and perhaps we can use that to design better therapies to specifically target the cell state. You can zoom out and do the same transcription factor analysis that I walked you through, and you can find the unique activity of certain transcription factors like NFAT half sites that have been shown by other groups. Okay, so in this section, I'm trying to give you a sense that we can now do this with single cell scale, right? And that we can pair that with orthogonal information about the cell, like TCR sequence. So what we're moving towards now is really expanding this suite of multi-omic analysis paired with epigenomics, right? So we've done it with TCR, and I've showed you that. We're now doing sort of single cell FACS sorting with help from BD, single cell barcoded proteins, either with AbSeq or CiteSeq, single cell CRISPR analysis with ATAC-seq, and then building a suite of analysis tools to analyze all that data. And our view is that it's nice to have the epigenomic information to really understand the cell state, but we really want to connect it with what everybody else is studying using cell surface proteins or targeted RNA expression. So just in the last two slides, I'll show you an example of some of these up-and-coming technologies. I'll focus on this paired CRISPR screening with ATAC-seq, but you can imagine the same type of analysis for AbSeq paired with ATAC-seq, right? So we're just doing high multi-dimensional analysis of many, many parameters. Doesn't really matter if it's CRISPR guides or antibodies, right? The logic is the same. So the standard way to do a CRISPR screen is to take a pool of cells, put in a pool of CRISPR guides, and then impose some selection, either cell growth or cell death, something like that, or maybe the expression of a single marker, and then look at what comes out of that pool. Okay, so our guides depleted or enriched to associate them with that particular selection that you put in. Okay, so this is great if you're studying cell growth. What if you're studying something that you don't have an obvious marker or an obvious selection protocol for? And so what you really want is to do this at single cell scale, right? So now you get your pool of cells, you put in your pool of guides, and then you measure what happens to every single cell with every single guide, right? And therefore, you remove any need for a selection, right? So if you have, for example, a T-cell exhaustion where you really want to study the epigenomic state, but you don't have a marker that tracks well with that phenotype, you can now address that because you're looking at every single cell. Okay, so just one or two data slides on this. So these are the types of things that we can do now with this setting, right? So now we're looking at, in a single experiment that takes about 2 hours or so, we can target using CRISPR, chromatin modifiers, transcription factors, even RNAs, and then do on the scale of 100 or 200 perturbations in a single experiment, and then get out, read out from each single cell that got each single guide what happens to the cis elements genome-wide, what happens to the transcription factors genome-wide. Okay? And the beauty of doing these sort of single-cell high-dimensional analysis is not really in the single profiles, not in the cells that got one transcription factor knockout or another. You could just do that in a bulk experiment, right? The beauty comes from, and again, this applies also to antibody staining, right? It's the same logic. The beauty comes from the single cells that get multiple things, right? So multiple guides, or multiple co-staining of antibodies or intracellular markers, things like that, and then see, how does that cell that gets multiple, in this case CRISPR knockouts, compare to what you would have expected from either single. Okay, so this is sort of summarized in this sort of cartoon here, right? That The x-axis is the expected epigenomic phenotype, and the y-axis is the observed epigenomic phenotype. So in a double targeted cell, if you just got what you expected from either single, so the addition of both singles, things would line up on a straight line, right? So the observed would be just the addition of the two singles. But what you actually see is many new interactions, right? So thousands of new regulatory interactions that are controlled by multiple transcription factors or chromatin modifiers that you don't see if you just knock out one or the other, right? So in this way, we think that we can really start to understand at scale with single cell resolution how different molecular networks, particularly in primary, this is done in primary T cells, how they operate in primary immune cells from patients. Okay? So I'll stop there, and I think what I've tried to give you a sense of is really the suite of genome technologies that we're developing to understand this non-coding genome, right? And we feel like this has really been a blind spot, particularly for immunologists, because you haven't been able to take these types of measurements in primary cells. And so now we're trying to develop tools to look at both the 3D genome conformation, so to give a background of which enhancer maps to which gene, and then take that analysis of accessibility to single cells, pair it with either protein or TCR information, and then develop platforms to do really high throughput perturbation experiments, all done in primary patient cells. Okay, so thanks very much for your attention. So of course it takes a village, so here are a lot of the people who contributed to this work. So I'm happy to take any questions.